Good afternoon, and thank you all for the opportunity to present my research. My research is based around the pretreatment and valorization of lignin. So a bit of project background. In recent years, there have been an increased urgency in finding alternative energy sources due to diminishing fossil fuels, as well as the impact on the environment. Transportation acts as the main driver for increased global oil demand, leading to a predicted consumption of 116 million barrels a day. This according to the Royal Society. These, amongst other issues such as growth in emerging economies, for example, China, have caused an acceleration in biotechnological research towards the development of a biorefinery system, which is capable of producing biofuels from renewable resources such as plant biomass. A number of feedstocks ranging from agricultural waste, such as straw from wheat milling, as well as marine cultures such as seaweed, have been proposed as an alternative source for renewable energy, as well as other essential fuels and chemicals. These types of feedstocks are classed as lignocellulosic biomass, composed of carbohydrate polymers such as cellulose and aromatic polymers such as lignin. So lignin is an organic aromatic heterogeneous polymer which makes up between 10 to 35% of most plant biomass. It's also one of the largest byproducts of second generation biofuel production. Lignin is made up of three polypropane units known as coniferyl alcohol, sinephyl alcohol, and p-coumaryl alcohol. These subunits are held together by a number of bonds, as displayed on the screen to the right. So the development of competent biorefinery processes demand the pretreatment of lignocellulosic biomass in order to break down the lignin polymer within lignocellulose, which is highly resistive, in order to release the cellulosic material that can be refined for various purposes. So some of the current implemented practices in pretreating lignocellulose biomass are physical and chemical methods. Majority of these processes result in inadequate separation of cellulose and lignin. It also causes the formation of byproducts, which inhibits the fermentation of ethanol, and also use high energy and chemicals, which cost a lot as well. Lignin can also be valorized to produce a number of hydrocarbon polymers used in different industries, such as pharmaceutical industries. For example, you can valorize lignin or depolymerize lignin to create vanillin, which is used as an artificial vanilla flavoring. So bacteria and fungi, they produce a number of extracellular peroxidases that have been implicated in the degradation of lignin. Although fungi peroxidases have shown to possess better activity compared to the bacterial counterparts, they are unfavorable for industrial implementation due to their instability at desired reaction conditions, such as temperature and pH, as well as limitation in carrying out bioengineering investigations on their enzymes. This has led to a lot of engineering studies in bacterial peroxidase investigating for improved activity. Dideoxyrizing peroxidase represents a family of heme peroxidases that are produced by a range of bacteria. Deeps are monomeric peroxidases, which confer a broader range of substrate specificity compared to other peroxidases. DYPs have also been implicated in the degradation of lignin through oxidative mediators such as manganese. So an investigation carried out by Mark Artman, where the dyp1B gene within Rhodococcus justi was deleted, resulted in a loss of activity towards lignin. Also, improved activity towards lignin by dyp1B was also observed in the presence of complex manganese, as we can see from the results on the screen. Also, a third group, Tim Boggs group, have also shown that dyp1B from the gram-negative bacterium Pseudomonas fluorescens displayed activity towards both manganese and lignin. These findings are supported by investigation into the role manganese plays in lignin degradation. It has been reported by a couple of papers that manganese can become, oxidized manganese can become chelated by organic acids, resulting in breakdown of phenolic structures within lignin. Manganese as Mn3+, by means of oxidative degradation of lipids, also produce phenolic radicals, which have been shown to oxidize the non-phenolic structure within lignin. So there have been some investigations carried out to improve manganese oxidation by deep enzymes through mutagenesis. So a study carried out by Singh et al. So in this paper, or in this project, displayed on the screen is the crystal structure for one of the mutants reported in this paper, where the manganese binding site was identified. The alanine residues highlighted was a substitute. Originally, there was an asparagine residue in that particular position. It was shown by Singh et al. That the substitution of this residue close to the binding site of manganese increased the resulting mutant's catalytic efficiency towards manganese by 14 folds compared to the wild type variant. So this work formed the main basis of my PhD. So my PhD, I aim to add to existing knowledge on the role DYPs and their mediators play in the degradation of lignin. Specifically, I aimed to improve lignin degradation by dyp1B from the gram-negative bacterium Pseudomonas fluorescens by site-directed mutagenesis of residues near the manganese binding site. So in order to carry out my aims, first what I did, I carried out some bioinformatics studies where I aligned the sequence of dyp1B from Pseudomonas fluorescens to the sequence of dyp1B from Ehibhatiomhan, which is the widely reported DYP enzyme in literature, as well as aligning it with some other DYP homologs that had high similarities. So upon alignment, I looked for regions of conservation which were verified by literature and the solved crystal structure available. So according to literature, all DYPs, with a few exceptions, share 3 conserved residues in the heme binding pockets. So these are an aspartic acid and arginine in the heme distal face, and a histidine residue, which is proximal to the heme ion. Next, based on the crystal structure of Ehibhatiomhan dyp1B, the multiple alignment, and a dyp1B homology module of the dyp1B binding site, which alluded to a histidine residue close to the binding pocket, I was able to identify the proposed residues involved in dyp1B's manganese binding. So I identified 6 residues, and of the 6, 4 residues were highly conserved in all the homologs I looked at. So that was 3 glutamic acids and 1 thyrine. This was consistent with the binding site of Ehibhatiomhan, as can be observed on the left. These 4 residues, along with the heme, work to bind manganese. This left me with 2 residues, a histidine at position 127 and a serine at position 223. A point to make, although these residues may not directly bind manganese, mutagenesis studies on similar residues at the same position in other dyps have led to improved dyp activity towards manganese, as was shown by Singh et al. Singh et al.'s work that I spoke about previously. The running hypothesis is that changes in these residues have an effect on the redox potential of manganese and oxidized manganese due to the proximity to the binding site. This is consistent with literature where a group found that using different ligands with manganese complexes had an effect on the redox potential due to various factors such as the residue's chelating structure, geometry, and electron donating effect. In particular, they found that the linear complexes stabilized oxidized manganese. So coupled with the solved manganese binding site of Ehibhatiomhan dyp1B, the multiple alignment, I decided to carry out the following mutations. So first I carried out histidine to arginine, and then I carried out a serine to asparagine, as well as a serine to alanine mutation. I also carried out a double mutation, which included a histidine to arginine mutation, as well as the serine to asparagine mutation. So upon completion of mutation, I designed and implemented a T7 LAC-based expression system in E. coli BL21. Expression vectors containing the different dyp1B variants were transformed into E. coli, grown and expressed in the presence of ampicillin and IPTG. Expressed proteins were harvested and purified via NINTA chromatography, assisted by a conjugated his tag on the protein. Next, I set out to characterize the resulting mutants, both in vitro and in vivo. First, I carried out some circular diachrosm just to visualize or compare the secondary structure of the wild type and the mutants, because what I didn't want was any subsequent changes as I record during my investigation. I wanted it to be due to the effect of the mutations rather than due to any changes in the secondary structure of the protein. And what I discovered from the circular diachrosm was the spectra produced for the wild type was more or less superimposable with the rest of the mutants, which alluded to the secondary structure being the same or similar for all variants. Next, I looked at some stability work. So I've looked at temperature profile. So for the temperature profile, the dyp1B and the wild type and the mutant variants were investigated at temperatures of 25 degrees to 65 degrees. All variants were most active between 50 and 60. However, the H127R and double mutants H127R S223N displayed highest activity at 60 degrees. So here I have measured some Menchel and Menton's kinetic parameters, comparing the mutant variants to the wild type using a number of peroxidase substrates. So the following parameters were measured. I measured Vmax, the maximum velocity. I measured Km, affinity of the enzyme to a specific substrate. I also measured Kcat, a measure of enzyme reactivity that detects how quickly an enzyme oxidizes or reduces a substrate. And I calculated Kcat Km, which is a measure of an enzyme's catalytic efficiency for particular substrates. So for ABTS, what I discovered was S23A variant was the most active variant with a 12-fold increase in Kcat compared to the wild type. For DCP, H127R variant was the most active. However, it was comparable to the wild type variant. There wasn't much difference. For manganese, which was the most important investigation, because the whole point of the project was to improve manganese oxidation, what I discovered was the S23A variant was the most active variant with a 20-fold increase in Kcat compared to the wild type variant. The increase in S23A's reactivity with manganese may be due to the substitution of the serine hydroxyl group. This may have acted, the serine hydroxyl group may have acted as a ligand, and then the change to an alanine may have changed the coordination state of the manganese center and perhaps stabilized the oxidized manganese. So next, I carried out some lignin assays. For Kraft lignin, initial velocity was recorded as change in absorbance over time due to the heterogeneous nature of the substrate lignin. I'm not exactly sure what of the products that were formed during this reaction because of the heterogeneous nature of lignin. However, I was able to observe a change in absorbance at 465, at a wavelength of 465 nanometer. From the data collected, it can be observed that the wild type and mutant variants displayed comparable initial rates of reaction without manganese. With manganese present, there wasn't a significant change in reaction for the wild type. So that's the gray and yellow bar for the wild type. There wasn't much change. There was a slight difference, but not significant change. However, of the mutant variants, all mutant variants displayed significant increases in initial rate in the presence of manganese, with an estimated 2.7-fold to 15-fold increase being observed, with the highest increase observed in the S23A variant. The S23A variant, consistent with the results from the manganese assay, was displayed a 5-fold increase in initial rate in the presence of manganese compared to the wild type. Next, I carried out an assay developed by Team Boggs Group that uses dinitrophenohydrazine, or DNP for short, to detect ketone and aldehyde products formed by oxidation of polymetric lignin. Again, as can be observed, S23A displayed the highest estimated product release with a near 2-fold increase over the wild type variant. So at the moment, I'm in the process of characterizing these mutants in vivo. I aim to express them in a bacterial host. In this case, I aim to express them in Pseudomonas putida and investigate the resulting strains for their capability of using aromatic compounds derived from lignin, as well as polymetric lignin as the sole carbon source. I also want to carry out some experiments to compare the effect each mutant gene has on the strain's ability to break down lignin and lignolytic substrates in the presence of manganese. And I want to analyze each individual strain for their metabolic products through HPLC. Thank you for your time and look forward to your questions.